going to start with the problem set. We're going to start with the first short answer questions. Aaron is going to begin. If we do not finish the entire set today, I will finish it the remainder on Monday, and the recording will be available for the remainder of the question. This set has slightly more questions, why Neuro has a little more content. So if we don't get through everything today, rest assured it will be finished on Monday, and I will post the recordings as part 2. So Aaron, please go ahead.
You want me to read the question?
Yeah, please read the question.
All right. You respond to a 911 call where the 65-year-old male patient suffered massive myocardial infarction on the left ventricle. He has AMS and presents wet and cold with cardiogenic shock. His vitals reveal BP 6854, heart rate 108, breathing 32 times a minute with SpO2 89% room air with positive crackles at the bases upon auscultation. After you finish your primary and secondary assessment, you decide to provide a vasopressor as part of your treatment. Based on your knowledge of receptors, which drug profile would be the first drug of choice to maintain his CO and hemodynamic status? State major adrenergic receptors and their functions, and what neurotransmitters are responsible for their action.
Yeah. So here, the drugs that I'm referring to, they're all found in your formulary, right? So we have dopamine, right? We have Norepi. So let me just circle. I'm going to circle the drugs. So I'm going to circle the doses. 1. We got this. We got this. So all these drugs, 1, 2, and 3, are found in your REMAC protocols. My question is, which one would you give as the first drug, and why?
I would give Norepi.
And why?
Because it has the greatest effect on the alpha-1 receptor and the lowest effect on the beta-1 receptor.
And why don't you want to give something that stimulates beta-1?
I believe because it could increase oxygen demand to the heart.
Yeah. So Aaron is 100% correct, right? So what you see here, all the pluses mean the most effect exerted. So let's say 4 pluses will mean it does more for alpha-1 as opposed to dopamine at this 5 to 10 dose. So we have a patient who had a massive heart attack. Obviously, his heart is not pumping as strong. It has increased myocardial oxygen demand. The other factor is you see that his heart rate is 108 to begin with. So his heart rate is already in elevated state. It's tachycardic. He's also suffered a heart attack. So we don't want to stress his heart any further. So if you look at your profile of the medications, right, you will say, well, how come we don't give epi? Right? Yeah, epi is going to cause alpha-1 response, but look at how much beta-1 response I'm also going to add to that, right? Someone who is already having issues with their heart, do I want to increase their heart rate to 140, right, or 150? I don't really want to do that. Why? Because I'm going to increase myocardial oxygen demand, as Aaron was saying, and I may cause further damage to them. I'm not going to help them. Actually, in the setting of heart attacks, in the hospital, they're going to give them beta blockers to lower down the heart rate, to reduce that myocardial oxygen demand. So the 2 drugs that you could give as your first line here would be the following drugs. Let me erase this. The 2 drugs that you could give is dopamine, right? And you could give norepi. So dopamine, here you will have alpha response, right? It's not as profound as norepi, but you notice the beta response is the same, right? So the beta response is not as elevated for dopamine. Epinephrine will not be the best drug of choice in this particular setting. Why? Because I don't want to increase the stress of his heart, right? Sometimes in extreme cases of shock where patient is not responding to a single pressor like norepi, we'll add additional pressors like epinephrine, phenylephrine, or vasopressin. Why? Because they have slightly different profile, or they may be working on slightly different receptors to cause vasoconstriction. So in severe cases of shock, we may add additional pressors to basically increase our effects, right? So here we see they say the answer is in the classic wet and cold shock, cardiogenic shock. This is what it means, CS, cardiogenic shock. The vasoactive medication you want to give is norepi or dopamine, and then they also add inotropic drugs. What are inotropic drugs? These are the inotropes, right? They cause increased contractility, so you see beta-1. majority of it is increased contractility. But the problem, right, with these drugs, initially they cause vasodilation. So if you look at the alpha, right, you see how it's negative? It means it vasodilates. So if your BP is 68 over 40, right? What did I write? I wrote 68 over 54. This is not the best drug to give initially, because initially I want to stabilize your blood pressure. I want to increase your vasomotor tone. Remember, I was drawing this diagram where you had the heart, and here you have the aorta, right, and the arteries. I want to vasoconstrict here, so I increase your vasomotor tone so that I bring up your blood pressure initially. So that's how they do it in the hospital. We'll give a pressor, norepi, right, to stabilize your blood pressure, increase your cardiac output, and mean arterial pressure. And then we'll add an additional drug, right, even though it's going to decrease the blood pressure somewhat, but we already have elevated the blood pressure, so we have some wiggle room to work with. Right. This is another important concept you're going to learn in your pharmacology, right? If a patient is in state of shock, right, all your medications for intubation, they will drop the blood pressure further. So all your drugs in MF5, medication facilitated intubation, they will drop your blood pressure. So if you decide to intubate someone and they still have, they're still somewhat alert, right? They still have ability to swallow. They're still able to speak somewhat, and you need to sedate them. Make sure you resuscitate them first to get their blood pressure up. You hit them with that lorazepam, diazepam, Versed, the benzo that's caused this GABA response. You will flatten their blood pressure. You'll kill them before you could even intubate them. So be cognizant. Blood pressure gets corrected before you add additional stuff to it. All right. Any questions about this?
I have a question.
Yeah.
Let's say you get Rust on like the cardiac arrest, right? You don't really know why they're in arrest. You just know that they're in arrest. You get Rust. Would it make sense to give push dose epi? Like, let's say you get Rust, you have a really low blood pressure. You give push dose epi and then hang like a dopamine drip.
So it depends where there's usually some sort of an etiology where there were shockable rhythms in the cardiac arrest. What was the 12 lead? So based on that, you could infer if there was myocardial injury or myocardial damage, and it was secondary to the heart. So I will say I will get to your question once you learn more of the pharmacology and EKGs and the pathophysiology involved. But there is a ways to distinguish. There's also ways to obtain history. So I would say you guys want like if you have A, you do B, right? And usually in in medicine that doesn't work. There's more to it. But once we learn some more stuff, there will be definitely I would say I will explain it to you more thoroughly. But in it's never the case of if you have A, you give B. You have to be more assessment work and more of diagnostic work, which you haven't learned yet. So I see, Ian, you have your hand up. You want to start with the first question?
Okay. Yep. Which of the following is not a function of the nervous system? I put D, direct long-term functions, such as growth.
That is correct, right? Yes. The nervous system is not responsible for long-term function. D is the correct answer. Okay. Number 2, I see Brian.
The blank nervous system is composed of the brain and spinal cord. C, central.
Central, correct. Central is correct. 3, we got Lindsay.
The blank nervous system controls the skeletal muscles. D, somatic.
Somatic is correct. We got Milan for number 4.
The autonomic nervous system provides involuntary regulation of smooth muscle, cardiac, and glandular activity. That would be
B. Autonomic is correct, right? So nervous system provides involuntary regulation of the smooth muscle, cardiac muscle, and glandular activity. Very good. 5, we got Curtis.
The basic functional unit of the nervous system is the neuron.
Neuron is correct, right? Yes. Then we have Priscilla. Would you like to go 6? Sorry, 9.
B, somatic neurons.
I can hardly hear you.
I think
if you said B?
Yeah.
Synaptic knobs. So actually on the axon, the cell body is connected to the axon hillock. So let me see if I can go back to that initial slide. That here you see, right, the axon hillock, and it's connected directly to the cell body here. Right. So cell body, what you want to remember, the cell body has all the cells in it, or organelles, the mitochondria, the nucleus, right? Anyone know what type of matter? We have white matter and gray matter. Oh, cell bodies are usually what type of matter?
Gray matter.
Gray matter.
Gray.
And axons being
The white matter.
White.
white
White.
matter, right? They're white because they have myelin sheath enveloping them, right? So this is what you want to remember. So it's the contents of the insides of the cell body has all these organelles, right? Very good. So let me go back to the questions. So axon hillock for number 9. Afterwards, we got Christina. Would you like to go for number 10? 12.
Neurotransmitter for release is stored in the synaptic neurotransmitter for release vesicle.
Vesicle, yeah, very good. So vesicles are made in the cell body, right? Why? Because there's nucleus. So cell, oh, perfect. Nucleus has the DNA material, right? DNA is turned into RNA. RNA is then forms proteins, right? We have transcription and then translation, and then the proteins is the vesicle that's now making its way to the terminals. The moment it reaches its terminals, it will be filled with whatever neurotransmitter that it has to, be filled with. Usually acetylcholine and the preganglionic cells. So the vesicle is 12 is correct. Let's do 13. We'll go back to the start. You guys can re-raise your hands. I see Lindsay, hand is raised.
The site of intracellular communication between neurons is the E synapse.
Synapse is correct, right? And you also have this specific cleft, right? Remember we talked about the synaptic cleft. That's where the neurotransmitter will be, but the synapse is where, right, the communication occurs. Very good. Next question is number 17. We'll have Milan.
Identify the structures labeled 5.
5 is here.
Would that be synaptic terminals? D?
Yeah, no, you're correct. Teledendria is higher up, and the axonal terminals are right at the tips. Milan, you're 100% correct, right? So axonal terminals are at the tips, and the teledendria is before that location. So let me go back to that. Yeah. So 4 versus 5. So 5 is lower down. This will correspond to synaptic terminals. So D for number 17 is correct. All right. We got Brian. Would you like to go for number 18?
Yeah. The structure labeled 2 is A. This was 18, right?
Yeah.
A, cell body.
Cell body. Yeah. Cell body is correct. So the whole thing is the cell body. Yeah. Number 22. We got, let's see, more people. Luisa, would you like to go for number 22?
Sure. A.
You saying A?
Yes.
Can you read the question so that we are everybody gets
Blank.
Mhm.
Monitor the position of a skeletal muscle and joint. I have A.
Yeah, proprioceptors. Proprioceptors is correct.
Proprioceptor.
Yeah. So 22 is correct. Proprioceptors is the correct one. Next, Jackson, would you like to go for number 29?
Okay. Sensory neurons of the PNS are C bipolar.
So usually in the sensory side, right? They're usually unipolar. Remember the sensory neuron I drew, right? We had the response. This was the cell body, and then went to the central nervous system. Let's say this was the CNS. This was the PNS. So usually they're unipolar. You have the touch receptor here, right? Remember we said it was sodium leaky channels, and you touch, and then the signal propagated like that to the CNS. So for number 29, the sensory neurons particularly are unipolar.
I have a question in regards to that. I thought it was bipolar because, like you showed, that there's the one sensor that when you like get a touch and then it goes to the central nervous system. Wouldn't that be two sensors then?
The actual. Let me go back to this drawing. I think this will be easier to understand. So this would be one pole where the impulse. You see how the impulse is propagating?
Yeah.
So from the sensory neuron, it goes to the central nervous system. It does not have two poles here. The sensory receptor is being the receptor directly, the receptor cell. So it's the actual target is it's not a neuron. It's not a neuron. The sensory part is the cell itself.
Okay, makes sense now.
Okay, it's just particular to the sensory portion of it. All right. We were at number 29, I think. Yes, unipolar is the answer. All right, now we go to number 36. Number 36, we got, if you guys want to raise your hands, I'm only going to pick for those who have their hands up. All right, so I see Crystal, 36.
Blank are the most numerous types of neuron in the CNS. Let's see. I know neurons are just a lot, but I don't know which one.
Which one did you just say before?
No, I know there's a lot of neurons, but I just don't know which particular one is.
Yeah. So usually it's interneurons as they communicate between each other. So interneurons is usually the most abundant type, or the most numerous type, as they communicate between other neurons. All right, number 39, we'll have Brian. Brian, would you like to go for number 39?
Sorry, can you hear me?
Yeah, Brian, you go ahead.
All right. Functions of astrocytes include all of the following except B, conducting action potentials.
Good, right? So that's not done by the astrocytes. The types of cells that we have, in the lectures videos, I go through it. I know I did not do this on this review, but once you watch the videos, I definitely go over it through that, right? So okay, 40. Let's go to Ari. Ari, would you like to do number 40?
Love to, not gonna lie. 40. The myelin sheath that covers many CNS axons is formed by, can be C, oligodendrites.
Oligodendrocytes.
Yeah.
Yeah. So that's, remember, right? Anyone recall for the peripheral nervous system? What was the name?
Schwann cells.
Schwann cells. Very good. Very good. All right, 42. Let's go with Sarah. Would you like to do number 42?
Yeah. Small wandering cells that engulf cell debris and pathogens in the CNS are called, I put D, microglia.
Microglia is correct. They're kind of similar to, your macrophages, like the big eaters, right? If they find the pathogen, they will engulf it, right? Phagocytize it, like engulf it, and right. with lysosomes and degraded or presented to the immune headquarters. So microglia, right? They are in the central nervous system, and that's their function. Very good. Next one, we got number 50. Well, let's do Helmi. Would you like to go?
Region of the central nervous system with abundance of myelinaceous axon constitute the white matter of will be A.
A, yeah. Myelinated axons is usually the white matter. We said the cell bodies was the gray matter. Yeah, very good. 51. We got, let's see, who else is raising their hand? Brendan, would you like to do 51?
Yes, I would. So regions of the CNS where neural cell bodies dominate constitute the gray matter.
Gray matter, very good. Yeah, so where cell bodies are gray matter, they have all the organelles, I should say. And yeah, that's where they found 54. Let's see who we got. I'm gonna start from the beginning, I think. Yeah, everybody won. I'm gonna start from the beginning again. I have Brian.
The neurolemma of axons in the peripheral nervous system is formed by E, Schwann cells.
E, right? You said E, right?
Yeah.
Yeah, Schwann cells is correct, peripheral nervous system. And by the way, what disease process demyelinates it in the peripheral nervous system?
Guillain-Barré syndrome, I think.
Yeah. Guillain-Barré syndrome is the correct syndrome. Number 58, we have Jackson.
58.
Yes.
The tiny gaps between adjacent Schwann cells are called gap junctions.
Yeah, so gap junctions are not found between the Schwann cells. What actually is found is the nodes, and there were. Remember, I was drawing this picture, and then I said, right. So let's say these are the Schwann cells, right? And these we have the nodes. And you want to recall what were the name of the nodes?
Solitary.
Nodes of breath.
Ranvier. Yeah. Yeah, nodes of Ranvier. And the reason why they're important to remember is that's where the actual sodium-potassium exchange occurs. This is what is allowing us to have the saltatory conduction, right? Faster conduction that's preceding. So nodes for 58. Let's do number 59. We have Sarah. Would you like to go 59?
Which parts of the neuron can conduct an action potential? I put the axon and telodendria.
Yeah, that is 100% correct. Axons is how the, right? So let's say this was our cell body, and this is our axon, right? The nerve impulse propagates through the axon, and it goes to the distal end where you have the telodendria, right? So telodendria being here. So all those are what propagated. Very good. Number 60. Let's see. Going down the list. I think I'm up to Brandon. Oh, I see Mr. Price. Mr. Price, I see you raise your hand. Good. Mr. Price, would you like to go?
Yeah, what's that, number 60?
Number 60, yeah.
The separation of plus and negative charge across the membranes creates a blank difference of voltage of B potential.
Correct. B is correct. Potential. Oh. Right? Difference. So the separation of plus and negative charges across the membrane, right, is the potential charge. And the the membranes, right? Specifically, I'm referring to is muscle and neural. So these tissues have the capability of causing action potential. Usually, the primary cations involved in this is sodium and potassium. So that's what you always want to be familiar with. All right, number 61. Let's see who else we got. Brendan, would you like to go?
Yes, sir. The sum of electrical and chemical forces acting on an ion is known as its blank gradient. The electrical chemical.
Electrochemical is correct. Electrochemical is correct. Electrical and chemical, right? So electrochemical. I think that's kind of an easy, easy one for you guys.
Right.
All right, 62. We got. Let's see, Priscilla. Would you like to go?
Sure. Which of the following interactions between electrical and chemical gradients does not lead to establishment of neuro-resisting potential? I'm actually not sure, but I'll take a guess.
Okay.
D?
D says potassium ions are attached to the negative charges inside the cells. So potassium are not potassium ions are attracted to the negative charges inside the cell. So this question says does not lead to the establishment of the neuronal resting potential, right? So if you have potassium ions attracted to the negative charges inside the cells, that can reestablish the potential, right? But what cannot, so you guys want to be careful, does not, right? So what does not is electrical forces that push sodium out of the cell. Remember, I keep saying that cells are bags of potassium in the sea of sodium, right?
Yes.
So electrical forces push sodium ions out of the cells. We usually have sodium rushing inside the cell, right? So answer B does not lead to the establishment of neurons resting potential. Let's see on the other questions. So which of the following interactions between electrical and chemical gradients does not lead to the establishment of neuronal resting potential? So let's look at A. Chemical and electrical forces both favor sodium ions entering the cells. Yes, they do, because remember we said the gradient for the sodium was, first of all, it was cells were bags of potassium in a sea of sodium. So there's more sodium millimolar, 145, outside the cell versus inside the cell. Plus, sodium wants to come in from the outside, right, to establish its equilibrium potential. And the equilibrium potential was plus 61 for sodium, or roughly about there. So A is true. Let's look at C. Chemical forces tend to drive potassium ions out of the cell. Yeah, that makes sense, because cells, again, the bags of potassium in the sea of sodium. So if the potassium leaves, right, it wants to establish its gradient equilibrium potential, which is about, which was a negative number, right, negative 80 something, 81. Potassium ions are attracted to the negative charges inside the cell. Yes, that is true. We have those exchange mechanisms with the leaky channels, potassium leaky channels. Potassium ions are repulsed by positive charges outside the cells. Yes, that's also true. So all those things are make sense. Potassium being a positive cation, it will be repulsed by any type of positive charge. So the only one that does not lead to the establishment of neuronal resting potential would be electrical forces that push sodium ions out of the cell, right? So this is the not question. I'm going to actually end the review questions at number 62.